Control deck, that is Hercules away.
Roger.
Control deck, that's Hercules past the transom.
Roger.
Control deck, that is tether all out.
Roger that.
Control deck, that's Atalanta away.
Roger that.
Hercules is dive.
This is how it begins, the not on my watch. This is just everyone's friendly reminder that no one is on SPL yet. Sounds good. And to lose your commentary over that.
Control, back deck. All stop 50 meters. Control's been sent to the van.
Roger that.
This is a voice slate for dive Hotel 2083 at 075000 mark.
Thank you, pilot.
And I'm on SBL now. Good afternoon, everyone.
Welcome.
Dave, the WCAs are powered up. Should get video in about 45 seconds.
Copy that. Thank you.
And let me know if there is, if they do not come up. I will push more buttons.
Dan, what's the first depth that we're going to?
I've been informed that the seabed is 700 meters.
Do you know approximately how long it'll take us to get down there?
21 minutes, 15 seconds. 21 minutes, 45 seconds. So somewhere around 20 minutes.
Great.
If you look at the Hercules pilot screen, if you have that on your KVM back there, I currently have the page up called Utility, and that gives a raw and parsed vertical velocity. So the vertical velocity at the top is currently 32 meters a minute and change, and that also calculates time to bottom. So it's now 70.
Awesome. Thank you.
Yeah, I need to turn on some lights. I got no light. It's getting dark and scary.
You're hauling the mail here. You want to slow down a bit?
No, you go faster.
No, you're going 34.
Yeah, you can do 34. No problem.
Dan, I have both WCA cameras.
Roger.
The lower camera still has all
You're
of
doing
the
37. I can't do this.
all the focus
This is no
assist
good.
and menu stuff
You
on
could
it.
do 35. Say again, Dave?
The lower camera still has focus assist, histogram.
Roger. Yeah, that's all Rachel. I'm not even looking at that thing.
I looked in the menus yesterday and couldn't figure out how to turn them off.
No, that's all Rachel in the background.
Okay.
35's no issue, Robert. We've been doing it all year.
Not on my watch.
Oh my God, it's been 2 minutes.
These are
Hour watch. We'll go in the middle. Do 33. How's that? Compromise?
Well, I'm way behind you.
so
I'll stop for a minute.
1, 2, 3, 4, 5. So if we go pip 5, it should say, no, I got it wrong. It's 1, 2, 3, 4, 5, 6, 7. Okay. 2, 3. So pip 3.
Sorry, I should have slowed down earlier.
Change that
I
to
didn't
the
see.
white.
Last time I looked at the delta, we were like 10.
Source ROV up.
There's my little eyeball. Okay, head it down.
So
I think Josh must have thrown an extra lump of lead on for you, Robert.
Yeah, that's kind of a concern. That's pretty fast.
We've been doing 35.
Yeah.
Yeah.
source camera
Ever since the frame rebuild, if you recall.
Oh, I don't drive down at 100%. So.
what happened?
Really?
Yeah,
What do you drive down at?
like 80.
Can you go adjust the camera? It's a lipstick camera just like this, right by there. I think she's got a monitor in front of it. This should be pointing down the
I've
line
always done 100.
by the data log.
Well, it used to be pokier. Seems like.
I'm just going to put the PTZ on the manipulator so I can keep an eye on it. Because it tends to salute if we're not watching it. Since we have Mike giving the winch much love, Mike and TJ, it's been great. It's actually rated for 60 meters a minute.
Yeah, but
Hercules.
I thought that was verboten, like going over 30. That was the word from the cheese.
No, he's totally into 35.
Yeah? Is that what he's doing?
I believe so, yeah. I ran Atlanta down at 60 last year. Dan,
What?
I ran Atlanta down by itself at 60 meters a minute the whole way, all the way down. No dramas. The bearings came apart, but they needed to be replaced anyway, so. No, it's all good for the most part. Then you can tell Oreo he has a free head now, and he can rotate around the center if he wishes. Thank you. You're also good for center rotate if you wish, Oreo. You're also good to pivot around the center if you wish. All right.
I'm not hungry right this second.
You're talking to them and Nav is talking to them and it can kind of get a little confusing, right? Yeah. Anyway, we got a good heading there. We got it.
So what's the scene here?
What's the scene, Jelly Bean?
What do we, are we starting a new one of these, or what are we doing?
Yeah, I did not do that. My bad.
So how do I do that?
If you look all the way to the left, there's a template. Hit the drop-down arrow. Right-click on the template and say duplicate. Then double-click on that and rename it to H2083.
H201?
83. And we've been out of habit, those gauge numbers that he calls out, I'll usually write them there. If you look at the previous pages, you'll see. But I can tell you right now, you can put...
Control shift, what? Plus?
Control shift colon.
What is it? Colon?
Yeah, control shift or semicolon. That one. I always get those two confused. That should be, you can just put 126666.
What do I put in here? Notes.
No, so we've been recording these on deck. So.
Oh, I see. Pre-dive gauges.
Yeah, so that's 12 and 06. 12, 6666 or 67, whatever. Dealer's choice.
Yeah, all right.
We're not going to argue over PSI.
Yeah, you went too fast, so I didn't get it all.
You can put 6s or 7s in there, Robert. Doesn't matter.
Yeah.
1 PSI. No one's ever going to know.
Res percent. So I can just put
100.
what the numbers are now, right?
No, new line, and then you can put in whatever you want. That's just an on-deck reference. We've been trying to capture that because there's been some...
When are we going to, like, automate this?
When we build a universal control module and hack the compensators to
shift.
Have a position sensor, and we incorporate Chris's software that records everything.
Well, yeah. Oh, so how does the data get collected?
I don't know. Why did that do that? Shouldn't have done that. That's good. You got to hold the jaw closed when you turn the hydraulics off because it.
Yeah.
There's a secret button here for if you want to read the gauges. This RMM, so that's res main motor. It zooms in on them, and if you turn the down lights off, the mid lights used to have enough. So RMM, and then it's thruster's craft term. But it's losing its brain and doing the focus thing. You gotta.
But now this is right over the top of where I want to be.
But I do this. Let me see. I go. I know what they are. So I go bang 666 and 366. Yeah. It's over 6, so good enough.
It's way over 6.
Yeah. It's fine.
Ah.
Sorry, do you want to see him again?
Whatever.
We'll get a real one on the bottom, Robert. I'm not worried about it right now.
I was happy I could see there for a minute.
Yeah, I'm gonna turn the lights on so they can see.
Right.
On bottom check. So we get a 50-meter check, and then I'll watch them on the way down, and then I'll get an on bottom.
I'll just wait till Rye gets in here.
No, we do the whole. So we do the 50-meter dance, and then the on bottom dance, and then I need to fly forward here.
Well,
Did the winch stop? What's going on? What's going on?
it's wide open right now.
Oh, I must have bumped the stick.
And
Sorry.
Sorry.
Stick bump.
when we get a, something
Easily
to see in
distracted,
water,
out of the
and
box.
we're shooting for about 80%.
Now that's definitely breaking the speed limit there. Look at you go.
Yeah. Come on. Yeah.
So we've got
Science.
WCA, whatever they are, upper and lower cameras problems. The wide angle one is grabbing freezes, like there's a network issue. The lower one has the focus assist, the histogram.
When we have just
center,
the
cross,
midlights, Robert, we
all
can
the-
have the downs off, and we still get light there, but we're, as you
Viewfinder
know,
stuff.
wounded at the moment there.
So I don't know how you get it out of viewfinder mode. Second try. I looked at this yesterday. Fine. Video devices. Camera. Look.
Are we close enough to the drop site, Lynette? Are you going to want to move back? Or can we come down here? Oh, Samantha. You did. I'm kind of loath to move the ship while we're coming down with this current, so if you're close enough for science. That's a half an hour move. That's an hour move. And this current sideways. So you're happy to, yeah, I know, we'll see how that goes. You're happy to acquire the seabed here then? You're happy to acquire the seabed here? I would, because we're screaming down with the wind, so. All right. Screaming down at the lightning speed of 10 meters a minute. No, we can move. It just adds another dynamic moving the ship while the winch is moving on the way down. So, and we have that current, so I don't.
Okay.
We can do it if we wanted, but we're going to be on the bottom before you would complete the move anyways, so. We'll be on the bottom in 6 minutes, so let's hold here.
Dan, I was was able to turn off all the junk on the output of cam lower. Finally found the...
are you doing that via the web interface, Dave?
Yep.
Yeah, we don't want to do that, matey, because when you open the web interface, it takes control of the camera.
Okay.
And that makes Triclops Wrangler mad. And when Triclops Wrangler gets mad, then the kilo views get mad, and it's a cascading.
Okay.
So
All right.
at the moment, Dave, what's happening with that lower camera is all Rachel. So she's got free reign there to do. She's doing some development work with it.
Okay.
So we're not displaying that up here. What we're doing is we're putting TherberCam on PC4.
Okay.
And then when I switch over to the dive special, I will push some buttons up here, and I will select PC4. Samantha should know how to do this. I'm on all my other navigators trained. Right now it's Herc GUI, so if you want to do some magic there. So you see how the upper video is now having grief?
Yeah, it's been having grief all along.
Yeah, so poking the...
I didn't touch that one.
Yeah, but poking that, like I said, so when you poke that, that makes Triclops Wrangler angry,
Okay, so
and
been,
when Triclops Wrangler gets angry, the whole
Wide
system becomes angry.
The wide angle camera's been grabbing freezes since it was turned on.
Roger.
Before I poked at anything.
Roger.
So. But
I'm gonna,
certainly, you know,
I'm gonna,
helping troubleshoot. I'll fix PC4.
yes, please. I usually ignore it and go back to this screen, and I get Rachel. Rachel! We got garlic. She's actively debugging and doing what Rachel does very well.
So PC4 should be fed with which computer?
Stocam Nuck.
Stocam Nuck.
Roger.
Copy that.
Oh, nice. Roger. Roger. Hopefully that'll be uphill.
Okay, TherberCam's on PC4.
Roger. So what I did there, Samantha, and for navigators, that's a really cool, powerful tool there. So on this layout, don't let me get too distracted, because the other thing that navigators are watching for is those Doppler beams to start. They'll go 1, 2, 3, and then we'll get 4 green ones. That means we're about ready to slam into the seabed. But what I'm doing, I have, so this is a graphical layout of that multi-viewer, and I currently have, I should have had picture-in-picture 3 selected, and so that's 1, 2, 3, as you see, 1, 2, 3. That's my destination, and if I use that as a source, then I get the digital still camera up there. It's yeah, also known as the TherberCam. It is, we're trying to get away from
that or the sexton, a housing name.
Yeah, manufacturer names.
Away from, yeah.
It's typical in science to name it from where it came from. So we refer to that as the upper camera. And we still refer to this as the Zeus, because we've always done that. And, but that one can be TherberCam or the digital still cam, or it's up to dealer's choice. We're going to be hurting on this one a little, because we're forced to use our downlight. So now I have, yeah, 3 beams. So typically at this point, I'll ask my co-pilot to remove the mechanical winch speed control device and go manual, and I'll do the same thing here with Hercules. So we're That way if we let go of it, things stop and bad things don't happen. Four beams. So Hercules currently has an altitude of 70 meters. I can guarantee you those are going to stay the same. That one's going to change a little. Right at 3, we're good. Means at 6, we're laughing. I'm getting a little uphill to Atlanta's left, and that is matching, jiving nicely with the at the grand plan of things. 40 meters. Meters. Visual on seabed, 20 meters. That's nice. I'm going to hold here, Robert, and I'm going to slide back on you. You want to kick your auto heading on for me if you don't already have it? Roger. You've done this before, have you? So I'm going to come to your west, which in theory is uphill.
Which direction we headed?
We're going to go, sorry, east. You know I get those backwards all the time. Dyslexic pilot.
Going east, so.
Eastish. That nav screen is being real slow to update, Samantha. Yeah, it's well, now it's behaving. Yeah, we've been on the seafloor for like 5 minutes.
I got it too, don't worry.
So I'm coming to the east here to, look, if you bring your head to the left a little, Robert, maybe that'll be, I think 075 looks uphill to me. Maybe look up the hill there. I'll call that good. Yeah, I think try 075, Robert. See how that looks. Yeah.
Can we confirm Norbit is operating?
Norbit is not on yet. Stand by. We have some bottom checks to do here, and I will turn it on when we get a chance.
A lot of crud in the water.
Yeah. So auto altitude double tap there. Auto heading. Ballast seems to be good. Good enough. Okay, science. Science is requesting something. What was it? Norbit. Norbit is on. I need to double check with the pilot before you press that button, because if they have auto XY on, Hercules is now trying to get back to Guam. I was watching. I'm gonna, where'd it go? Where is it? It didn't take. I'm gonna hand it over to Josh. He's sitting here patiently.
Rachel, if you're listening, or whoever else might know, the Norbit is not coming up on the Norbit computer. Oh, there it is. Is that, or no, you pulled up MB proc.
Okay, I think ready for you, Dave, to white balance?
I'll turn out the
Copy
lasers.
that.
Lasers off.
Tilt up just a little bit. Ugh. You want that more in the white?
Yes, please. Close to center as you can. I think I can zoom in a little bit. No, I'm zoomed out a little bit.
You're very quiet, Dave.
I am not normally very quiet, but you should be able to turn me up.
I cannot turn video up.
Huh.
Not here. Can't turn video, bridge. A few of them I can't. I have no
I
control.
can't hear you either.
Yeah. All right, well, I'm talking on SPL, and so.
Okay, now I can hear you a bit better.
Okay. All right, black balance first. Herc's going to black right now. I said now. I said now. There it is. And white balance. And we're happy. And save that. Okay, white balance is good. Thank you.
What is it?
Thanks, Dave.
That's the WCA upper.
Just gonna move the Orion out of the Thurber cam view there if I can. Oh, no, PRV right here. Yeah. And if it's easier for Oriole to have the center rotation at the center of the ship, that's fine too. We can deal with that change in the aft. Hey Dave, can you double check the Hercules is full wide, please? Oh yeah, okay, we're like slightly pushed in. Okay, awesome, thank you. You think are we good, Bob, from your perspective?
Yeah. Swell.
Swell. Okay. I think we're ready. Dave, are you good?
It is good.
Awesome.
I'm better than swell. I'm peachy.
Yes. Sure. Eastish. Good little sponge to start out with. Yeah. Looks like some neat shadows there.
All right, so we're rolling now. We're good to go. All right, so for people tuning in on Nautilus Live, just to give you a little bit of info on what we're doing some transects of this area. Right now we're at a depth of, what are we at? Just over 700 meters.
Yep, 735.
And we will get shallower and shallower as the dive goes on, and ending at, looks like, just 175 meters deep, is that right?
I believe that's correct, yeah.
Yeah, cool.
Yeah, for those watching now, we're off the west coast of Palau, off the western barrier reef, an area that's known as Shark City, which is a famous dive site. And there's a ridge, a submarine ridge that's very poorly known, which extends out about 2 kilometers from the shallow reef at a depth of around 180 meters. And we were at the outer end of that, down the slope on the western side, the side that faces the deep ocean, and we're right now at 730 meters, starting to transect up the slope to the very end of the ridge, and then we're going to move to the south along the top of the ridge. And a lot of interesting things going on down here. A lot of activity, bioturbation and such in the sand here. It's a mostly sand slope with these, here comes an outcrop of something with variety of invertebrates. We got some black coral whips. Can't really tell it.
Could we do a slight zoom in, please, Dave?
And there's some shrimp,
good.
some stylasterine corals, that pink on the left of the view. Nice shrimp. We zoom in on the shrimp just temporarily. Just
Sure.
get a good shot of it.
Yep.
Yeah, good. Oh yeah, lovely. And yeah, so that's a black coral whip to the right hand of the screen. Gosh, there's another nice stylasterine up in the upper left corner, another black coral. Fairly common sorts of things that you find at these depths in Palau. Look like some little yellow sponges maybe. They're on the rock, small ones, or greenish, not yellow. And off in the background there behind the rock, we see a purple soft coral, which is well known. Got a sideroid sea urchin with the heavy pencil-like spines.
Oh, come wide. Thanks.
Sticking out. Interesting stuff. So the, sorry, can you zoom in, see that white thing on the stalk, kind of in the middle of the view?
You can use that.
There we go, yeah, okay. Can you zoom in on it?
On which feature there, Pat?
It's that upper part of the view, the rock. Yeah, here we go.
Yeah, there we go. Awesome. Got it. All right.
Okay. Now, if I'm correct, I think we're going to find that that is a type of benthic ctenophore, which is ctenophores are comb jellies.
Zoom in, please.
But usually they're pelagic, but this is a, nope, I'm wrong about that. Not what I thought it was, but some kind of looks like a stylasterine coral. Anyway, you see a lot of sediment in this area. This is all, sorry, right there.
Sure.
Yeah, this sediment is, usually produced in shallow water and is being brought down the slope slowly. So you have shallow water production of the sediment up on the reefy areas that are shallower, and then it comes down the slope here into the deep ocean eventually. But, a very, very sediment-dominated environment. Yeah. So that in the upper, there's a white ball on a stalk. Can we have a close look at that?
Sure.
That looks cool.
Hey, we'll zoom in, please. If we want, do we want lasers on for scale, or no?
Okay.
Probably.
Looks like a sponge.
Yeah.
Sounds good.
Anybody want to vote to collect that? I can't.
Okay, well, come on, please. Is that something we want to do? We need to stop the ship if that's the case.
Just a second. We'll...
Sure, we can hang out here.
Lori.
Layla, what station is Pat at? Right. Okay, thanks.
Can you zoom in, please? Okay, we'll come
I got the Synaphobrankid. Cutthroat eel.
Should I just keep on moving? We might see some of those sponges again, maybe? I don't know. I haven't stopped the ship yet, so if we're going to sample them. Sure.
I think they're discussing.
Ah, okay. There'll be likely more, I would bet, but I don't know that for sure.
I don't know. Okay, so hearing that they would like to collect that sponge and stop the ship for it,
Okay.
if that is possible still.
Sure, we can stop the ship.
Oh, okay.
I just got to find it again. Oh, there we go. I'm going to be a little bit under Atalanta, but might be a snip. Snip and drawer, probably.
Yeah, I think they're discussing how to collect it. Yep. Yeah, it seems small enough. We could.
Yeah, just grab it at the base.
Sorry, I think I'm realizing that you were not on SPL. So we were hoping to take the whole sponge,
Yes.
snipped from the base as a voucher sample.
Yeah, you might actually try, sticking your manipulator claw a bit into the sand.
Okay.
And grabbing that. There might be a base anchoring it to the sediment.
Okay. Do you want me to do that, Bob? I can do that.
An ophiuroid coming up into the view. So we're at 726 meters.
Do you want to zoom in first, Dave?
Yeah, sure. That sounds great. Yeah, that's
Yep.
beautiful.
Got some, looks like there's a zoanthid on the stalk.
Yeah, something there.
Yeah.
Comes with the territory, I guess.
Okay, I have good
Okay.
shots of that.
Come wide, please.
Yep.
Will you zoom in a bit, please?
Yeah,
Okay, we'll come wide, please.
Oh, that's lovely.
Yeah, that is pretty.
Yeah, that's a beaut.
All right.
See if it's got a base to it there. You know, things like that anchoring out here in the sediment, it's really difficult because, you know, they usually need a rock or something to attach to. So it's going to be interesting to see what's on the end there when it drops it. Doesn't look like much.
No,
But,
no, probably didn't get the full.
yeah, it could
I
be.
can see
Or
little
just,
threads. It's kind of hard to see.
You know, you'd be surprised what these things can attach to.
Yeah, one single little pebble.
Little pebble, just a small pebble is sometimes enough. So that's a great collection.
All
728 meters.
I believe number 1001, nothing was collected while I was at dinner, right?
Yeah, so sponge.
Yes, 001, yep.
Stalked sponge. Great.
Pat, we have a question from a Nautilus Live viewer about how these sponges anchor themselves in the sand. Do you know kind of what that mechanism looks like?
Well, I would imagine, you know, down here, you know, sponges can have larvae that swim. And so you have a swimming larvae that's been produced sexually, and it's swimming around and it's looking for something solid to attach to. And so if they find you know, literally a pebble or a rock, you know, they can attach to that and start growing. Some of them choose the right place.
Zoom in, please, Dave.
Some of them choose the wrong place. That one obviously chose the right place.
Is that thing still hot, Josh?
So they look for something they can attach to. Just
The
fine-grained sand is just really not good enough.
you can leave it on.
But
I'll
even if it's just a
move
little thing, they can
I'll
work with
just
that.
move the. I'll just do this. Sure. It's up to you. I don't care. Yeah, I've been doing the same.
No.
Comms is switching out. Alice is coming on. See y'all after dinner.
See ya.
Oh, yeah.
No, that's a very common shallow water shell. These shells can roll down the slope too from shallow water. It's got a hermit crab in it, almost certainly. But okay. Yeah, that looks good. Can you zoom in any more? Just yeah. No, on the sponge.
Or the...
Okay, yeah, if you can get a little bit closer, it'd be kind of interesting.
No worries. Roger that. Coming away.
Why you want to insult Jacob like that?
Okay.
Full wide or is this good?
I did the same earlier. I called Samantha Lynette. I didn't realize they had changed out. Laser focused on something else.
Roger that.
Yeah.
That's a lovely view. Yeah. If I was diving, I would reach out and touch that to see if it was soft or hard. But
we do that sometimes. Yeah,
well,
a little wave.
sure. Yeah. We're not going to want to collect it, but knowing whether it's something hard or if it's soft, being a sponge would be interesting. Oh, okay. Oh, that's well. That's good. We can see it's got polyps on the one side. Sorry about that. He'll grow. You had one, now you have two.
I barely touched it.
Yeah, no, that's the way it goes. You know, yeah. Interesting. You know, it's probably a unidirectional current factor there. The coral is polyps are only on one side. It looks like it's bent to probably like a tree facing away from the wind.
So we have the capability, Pat, to recover that piece in our suction sampler if you wish.
No, we can't pick that up. Okay. Thanks anyway. But
there's other coral, I think, just off to the left.
Yeah.
It's a matter of the species of coral that we cannot collect.
Yeah, no, we can just start moving up more.
Okay, we can zoom in on that one, please.
There was a little...
we could spend 8 hours down here, and I'd be very happy.
We'll do a quick zoom on this one too, video, please.
I want to lodge a formal complaint with the scientists. I was promised cliffs, sheer cliffs, not mud.
Yep, yeah, we'll go for zoom. Do a quick zoom on this one, and then we'll get moving.
Oh.
Probably Victorgorgia.
Appears to be purple.
Yeah, I was trying to think of the name of that genus, Dictogorgia. Yeah, some brittle stars. Cool.
Great. Okay, we'll come wide.
Coming wide.
And should we be moving on there, Pat?
Yes.
Okay. Start up the ship. you.
What? Can you zoom out on HIPAC just for a second, Lynette? I didn't get a chance to glance at it earlier, my bad. Do we have pathing lines on this one, or? Come up a few meters, Josh.
Sure.
Got a black coral, looks like.
Yeah,
Maybe
that's a nice one.
bathypathes.
Feathery black coral. That are.
So I'm no expert on that, but it looks like it doesn't get steep till halfway between waypoint 1 and waypoint 2. Is that accurate?
Yeah.
Mm-hmm. Steeper. Yeah, they look pretty close. Okay, thanks. That's all I need to see.
So you see those tracks in the sand there? I mean, there are things, you know, crawling over the sand, probably hermit crabs and other crustaceans feeding on detritus coming down. Very active bottom, very live.
I'm going to swing out of Renta.
Yeah, I think we're going, what is it, 088 or something? 080, roger.
I'm 045 at the moment. I'll hold it there for a while.
Roger.
Yeah, if you see those, there are white mounds in the sand there, kind of with a, looks like a landslide going down from them. Those would be from infaunal burrowing creatures that are processing the sand and then expelling it out, and it makes a little downhill
There's something.
slag heap, so to speak, like they're mining.
What are some examples of those creatures that would do that?
Things like ghost shrimp, calcium acid shrimp, which are abundant in shallow water, and these are probably calcium acids. They're all over relatively shallow ocean. A whole bunch of other things. There are burrows of things that we see, at least in shallow water reefs, but we don't know what makes them. Just very, very strange stuff. Infaunal creatures are kind of weird, generally, and not that well known. Oh, that's lovely. That's like a primnoid gorgonian.
Yep.
Lovely.
Let's do just like a partial zoom in, please.
Roger that. Going in halfway.
Yeah.
Some paramerissid, maybe?
That it looks like one species we had before, we know before. Oh, that's had a myctophid that just went by. Lanternfish? Nope, no, it's not. So.
Nice, big eyes. Oh, yeah.
I forget the genus in a second, but it's a cardinal fish derivative. So we're at 713 meters. Epigonus, that's the genus of that fish. Epigonus.
Epigonus?
Yeah, I'm pretty sure.
What do you got going on PC 1, 2, and 3? Can I put Ross Brock on one of those?
What was that?
What do you got going out on PC 1, 2, or 3?
Looks like a farreid sponge, maybe, and a crinoid.
Crinoids, yeah.
Yeah.
Let me look.
Oh wait, I can answer that question myself, sorry.
Yeah, I got some zoanthids growing on the gorgonian there, the little yellow blobs.
Pat, what's the orientation of the fan?
Well,
The
they
coral
usually
tell us about the currents.
orient perpendicular to the current, the mean current.
Yeah, Herc heading is pretty much north right now.
I'll bring Atalanta to the north as well.
Okay, we'll come wide, please.
Coming wide.
That is massive. That was very cool.
Yeah, really big primnoid.
I guess in the last 2 cruises, you had a lot of primnoid gorgonians down fairly deep.
Yeah, we did.
This is, it's pretty deep, but it's not really as deep as they go. It's pretty interesting. They start showing up at about 250, well, about 75 to 90 meters in cool water areas, and then they get more abundant down 100, 120, 150 meters, but they go a long ways down. Well, these are interesting rocks. They obviously have probably rolled down the slope from shallow water. Are they basalt, or are they carbonate rock, limestones? Can't really tell from here. It looks awfully basaltic. It looks dark. We don't know really that much about the geological origin of origin of the ridge, but we feel it's probably made up of basalt from, you know, a volcanic eruption literally millions of years ago. And so, you know, it would makes sense that these are basaltic boulders. That look like that same sponge, those 2 sponges? Yeah, good. So those are 710 meters. That's a great sponge. Beautiful. Glass sponges, you think?
Yep. Yeah. Stalked glass sponges. Not sure exactly what.
Nice crinoid there.
Other sponges in the crevices.
Yeah. Oh yeah, they are glass sponges.
Okay, well, come wide, please.
Going wide.
Yeah, nice.
Coming up.
Do you want to keep the ship moving?
Yeah, I think so.
Okay. Bridge nav. Can we have 30 meters 085, please? Thank you.
Come on.
Can we zoom in, please?
Roger that.
That's a nice sideroid sea urchin. In the current. That's an interesting gorgonian. Yeah, it's dead. Might actually be it could be a hard coral. Kind of looks, who knows?
Okay, we'll come wide, please.
That's a nice little coral there on the left side of it.
You said isideroid urchin?
Yeah, pencil urchin.
Pencil, yeah. Oh,
Our
big sponge right there.
Thanks, Josh.
Well, good. I'm glad we collected that one because it appears to be a relatively common species down here, and now we'll be able to identify it. as the people listening, you should know that sponges are really tricky to identify. There used to be virtually nobody in the world who could identify sponges, and there were a tremendous number of undescribed species. The last couple of decades, there have been a lot of young people joining that field and advances with DNA and everything. So now you can, identify many of these things, and it's a very, very active area, taxonomy. So we have if it's not a known species, there are people who would love to describe it. So all you need is one specimen, and we'll find out what it is.
At least the early one we collected looks a lot like these hyalinema sponges. It seems like it's common to have the zoantharians on it.
Huh. Yeah, we're looking at
hyalinema.
Nema. Yeah, it could be the genus at unidentified species. So I'm sure it'll be really interesting to put it in its proper place.
Yeah, and always good to know the range of...
Yeah, depth range, geographic range.
Current's kind of kicking, huh?
Now, I'm impressed with, Hercules and Atlanta's ability to work in these conditions. It's very impressive.
That's all Robert and Dan and Lynette and Jacob in the front row, by the way.
It takes a team, as they say. You guys are doing a great job.
Dave, you want to zoom in?
Oh,
no, it's not Dave.
Eric, you've got...
Video, can you zoom in?
Going in. He'll be back shortly.
A nice little urchin out there.
Edge of your leash there, Robert.
Sure.
Another
Feel the tug.
Snaphobranchid, right?
Yeah, looks like it.
I used to work with a lot of ichthyologists who described eels, and you got to be kind of weird to work on snaphobranchid eels.
Lynette, can you?
Somebody has to do it, though.
That's what I want to see.
A rat tail flying by.
On the boat.
Floated by.
It's been doing this lately with the missing lines. I don't know what's up there.
Oh, that's only on your screen.
Oh, yeah.
Interesting. Do you want me to move up this way? Can you see my cursor?
Yeah. No, it's up to Robert. Robert's driving.
I can come back that way. If we're not on a particular course here, I'll just come over to you.
Okay.
We are. We're looking for a wall.
Can we zoom back out?
Going out.
Yeah, the current's ripping.
Yeah, we're just crossing the 700-meter barrier level.
Oh, there was a sea cucumber down there.
Coming back up. Where did you go?
We have a couple questions online about classification and taxonomy. What are the parameters that are used to help define species of sponges and corals?
I'm going to swing Atlantis heading around to your heading. Okay, Robert?
Well, you know, every species, you know, that is described has been.
I'm good. Yeah.
Been given a binomial name by, ideally by a taxonomist who has studied the morphology of the creature, and you know, people have been naming organisms for hundreds of years now, and you have to. Part of the problem is being really careful about.
Coming up a few more meters, Robert.
A new species, or is it something that's already been described?
Looks like we're going upslope a bit here.
Yeah, museums have collections of preserved material, just like, you know, specimens are being collected here. Those specimens are deposited in a museum to be kept literally in perpetuity, and then a single individual, say like that glass sponge we collected a few minutes ago, if that is a new species, that single individual could be designated what's called the holotype, and that's the name, whatever it is attached to that single specimen, and then that name is applied to that. The taxonomist does a very detailed description using various characters.
No, you could definitely get rid of that, because I don't like it.
It's floating by right there.
From the organism that's published.
Video, can we zoom in?
Yeah. Anyway, they publish the name, they publish a description. Oh, wow.
It's so little.
No, it's a jellyfish.
Jelly, yeah.
Oh.
Anyway, so I'm not sure that answered the question. We've gotten, you know, dragged off by this jellyfish drifting away, but you know, they use a lot of detailed scientific knowledge and study the literature over hundreds of years to know whether somebody's already given a name.
Going wide.
Worst comes to worst, you can actually refer to the original specimen that was tied to a name, and museums will let you come and look at it. They usually don't send out what we call the holotype, the original, but that worker can go to the Paris Museum or the Smithsonian that hold these specimens and compare the one they think might be a new species or might not with the original thing that's tied to the name. So if it's different, then they'll name it themselves. If it's the same, well, they just maybe have a geographic record or a depth record, or can expand the knowledge of that particular species at some level.
So it's a combination of both physical characteristics as well as DNA studies?
Yeah, I mean, DNA is the, you know, people use DNA now, going back to Linnaeus, you know, hundreds of years ago. People didn't have DNA, and of course now, looks like some more epigonus. If you have specimens, you can do DNA, and it's really a wonderful way to distinguish things one from
down
another.
In the data lab. I don't know exactly who.
Virtually every study, every taxonomic study today would be using DNA in terms of distinguishing the species one from another. Yeah, it looks like epigonus again.
So would you like, Pat, to generally chug along forward and zoom on things as we pass by?
Sure,
or?
yes, yeah, we should start moving upslope.
Yeah, are we looking for anything in particular, or?
Well, I'll know it when I see it.
Okay.
A nautilus?
You know, we never know what we're going to see next, so...
Okay, I just didn't know what to try and concentrate on as I'm going here.
Yeah, new species.
Okay.
Yeah, find the new species, Bob. Find them.
Or find something we don't know occurs and pull out. So.
Well, if it's something I've never seen before, I guess.
Yeah, that would mean something. If Bob's never seen it.
What are those on the...
Yeah, are these sea pens,
on the
or?
Left? Yeah.
Can we zoom in?
Yeah, let's go on the left there.
It looks like maybe small, that one looks like a sea pen, but all the other ones look like small hyaline sponges.
Yeah, that's a sea pen, right?
Or actually, I see bamboo notches on it. It's probably it's an unbranching bamboo coral. Yeah.
Yeah, but over there to the left and up there was some.
Can we zoom back out again?
Going out.
Yeah.
I can take it over here if they don't have it. I just didn't want to grab it.
Yeah, all those on the left.
There, are they different?
They look all the same, and so they're different from the Isidid. I think they're a sponge with zoanthids.
Yeah, it looks like our sponge on a stalk again. Maybe if we get a closer look.
Can we zoom in again?
It looks like zoanthids on the stalks too. Oh, that's great. If they're, it's really a common. Oh, that's wonderful. Yeah.
Yes, makes us feel better about taking the other.
Yeah, no, they've got a great record of that species from Palau now, and you'd be surprised how many of these things. Down here, the temperature is cold, it's dark, and it's cold and dark everywhere in the ocean. In the tropics and the temperate areas, and north and south, so that. A lot of these deeper water things, they have really broad geographic ranges.
Good there, Bob. Thanks.
Coming wide.
They're not tropical in particular. They're just found everywhere in the deep, dark, cool ocean. That's wonderful. Look at those guys. Wow, great.
Coming up.
Okay. We are at, where are we at now?
I'm going to zip ahead a little bit here.
All right.
Can't see.
Will a DVL give us a measurement of seabed current?
Negative. Not our DVL.
Yeah.
We can.
I can just go dead stick.
Yeah.
See.
Our technique is to let go of it and see how fast it pushes Hercules.
Yeah. So. Just on that last sponge, it's 690 meters, so.
Yeah, we can see how far up it follows us.
Yeah, where it ceases to be common, or we don't find it at all. And then, we'll correlate that with the temperature down here. One of the interesting things we've learned in Palau over the years, and this is mostly from physical oceanographic work, where they send out autonomous sea gliders, which actually go up and down from the surface, usually to depths of about 1,000 meters. Is we get a picture of, very detailed picture of the vertical distribution of temperature, and with changes from El Nino southern oscillation between what we call El Nino, La Nina. Now in Palau, La Nina is hot water at the surface and down relatively deep. El Nino is cool water and down fairly deep, and we can actually end up with an area like this, 700 meters deep. The temperature's not always the same. It can vary over probably 3 or 4 degrees centigrade. Now right now we're looking at about just a hair over 6 degrees C down here. So it might, El Nino, La Nina, you might vary between 6 and 8 degrees centigrade. The range of temperature over the ENSO factors, it gets larger as you go shallower, like 500 meters, 400 meters. But once you get down to about 1,000 meters, the temperature literally never changes because of ENSO, except by global temperature changes. It's very long term, but
sorry,
the stability in temperature down deep is really remarkable, and not so stable up here.
Flew over some rocks there to your right. Orbit picking them up to your right.
Some on the sonar.
Yeah. That bamboo coral again.
3 sonars.
Yeah.
Dan, what computer are you on with Norbit?
Rosproc. You should have that back there on your KVM as well.
Okay.
I don't know how we ever lived without it. It's a beautiful instrument. By the way, I wanted to plug, someone back there had asked about being able to sense current with Hercules. We've had many scientists request that, so if our audience and donors are listening, we would love an instrument on Hercules that could tell us what the actual seabed current is.
You know,
or on Atalanta. We'd love to have one on both.
Yeah, so just look at those tracks on the bottom. All that, disturbed sand, that's all from creatures crawling and dragging their shells, and just a remarkably, live bottom down here. Most people don't get very enthused about sediment, but it's important. It's covers most of the ocean, and it's great to see that things are doing things in the sand bottoms here.
Scientists always say that when we have mud instead of rocks.
Yep.
Yeah. You
Sorry,
can
Pat,
get enthused about those too, though.
Oh, yeah.
We saw some, on the last expedition, we saw these really strange, they were giant mounds, probably the width of Hercules, so maybe, definitely over 1 meter wide.
Really?
And I would say they were 300 millimeters tall, maybe a foot tall, somewhere around in there. And we could see no signs of bioturbation on them. And we tried to take a core sample in one, and we're unable to recover the core because it just, kind of pooped out the core sampler. But what we did see is a lot of kind of stratification of the layers in the mound, and we were never able to determine what was making these mounds. And we came across quite a few of them, probably maybe.
Really? What sort of depths were those at?
Oh gosh, I can't.
you
Sorry,
zoom on
Pat.
the
I don't
sponge while
pay
we
attention
talk?
to the depth a lot.
We zoom in?
Yep, good vision.
Yeah, well, we'll have to look that up. Were they like what we call volcanoes? A conical mound with a little pit at the top of it, a little crater?
Was no crater in the center, nothing to indicate any biological forms like, the worm that comes out and flails around and or some of the typical, excavators that.
That's good.
Yeah, it's just helpful to do
was a
brief
huge
zooms as we
mound.
fly by on stuff.
Reminded me of the gopher mounds in my front yard. With no sign of life, just a big pile of dirt.
Yeah, zoom out.
Yeah, we'd have to just check that out. These things, the ghost shrimp, produce big mounds like that in shallow water reef areas, and could be doing the same thing down here.
It stumped both of our resident experts, both Brian Kennedy and Steve, our deep water coral experts. Yeah, we couldn't see any signs of, the bioturbation you would see when things are digging around and throwing dirt out. It was they were really layered.
Yeah. The ghost shrimp, they live in the bottom. They have extensive buried burrow systems, and they pump water through those systems, gleaning out organics. And
coming up with you.
The mound they produce is actually the result of them pumping water and sediment back to the surface. And it is literally like a volcano, except it's not lava, it's sediments and water.
But you would see
and usually there's a little conical pit at the top, which is the crater, but not always.
Yeah, maybe that. And they're that large, like over 1 meter wide?
Yeah, the ones in shallow water certainly can get that big.
Maybe the sediment just filled them in.
Yeah, we should have a look at those. Maybe we'll see some on this dive.
Yeah. Look at Norbit painting that beautiful rock for you, Robert.
Yeah, it's cool to see.
Nice.
The eye of Norbit sees all.
Can we zoom in
Yeah,
again?
Eric, I don't know if you wanted Norbit up at all, but you can now pull that up on Ross Proc on your left one.
Sorry, what's that?
I was just telling Eric where he could pull up Norbit.
Oh. We were trying to look at something.
Nope. although it looks like there's a primnoid there maybe. And a pencil urchin again.
All right, zoom back out.
One way.
No. Yeah. Yeah. Okay. Next one. The next target. That one, well.
Just gonna pan your bubble cam there to look at the other gauges real quick.
I don't care about bubble cam.
Gauges are good.
You can have it.
Well, you'll notice that gauge checks are magically exactly on the hour to the second later.
Really?
How's that possible?
Looks like some nice sediment slumping
really
there. That
staying
little
on top of things.
little
That's
tiny
called
ridge.
copy paste, my friend. Gauges are good. I do change the seabed depth though.
So does it help you, Pat, for us to like quickly zoom on things as we pass by, not long zoom while you're narrating other things? Lori's nodding yes. Okay, so Lori would like, as we're passing by, to just focus on briefly on little things so we can ID better.
Sure, yeah.
Like what kind of little things?
Like that pink one on the right.
The pink one.
Yeah,
that's Chrysogorgia or something like that. I don't know. Yes,
what about
that,
that?
yeah,
Look at that
perfect.
fish up there, that's different.
To help
Can
that
you zoom in?
Robert got closer, he's light years away.
Yeah, it looks like a Chrysogorgia. Chrysogorgia crises, maybe?
A Galatheid in there, right?
Yeah. Yeah, so the Chrysogorgids usually have a squat lobster friend.
I think we'd like to collect that one.
You want to collect it? All right.
Is that definitive? Yeah?
Yeah.
Go ahead. Okay.
We'd like
all right,
the
they
whole,
would like to, confidence, we would like to collect that.
Yeah. You know, we need the whole specimen.
Okay.
Yeah. I'm just getting some
And
Copy of the net.
more pixies.
Hold position, please.
I know
We're at the
a squat lobster taxonomist at the Smithsonian who really appreciates when we
Can we
try
hold
and
position,
collect
please?
the squat with the
Thank you.
coral. Maybe
So-
we'll get lucky.
We're 665 meters.
Wherever, on the seabed. You can zoom in on it, I'll come in with the upper camera. So you can land and get some pretty close-ups while I get the minute out.
Who's your taxonomist at the Smithsonian for
Paula Santiago Gomez, maybe that's her full name?
It's, yeah, you figured out the buttons already.
Oh, there's another
Get a tight zoom.
squatty back there.
Come in.
So are those zoanthids or polyps, the little white
Those are the
pink
polyps,
balls?
yeah.
Yeah, they look like zoanthids, maybe we'll see. I forget the specimen.
How much of this do you want, Pat?
Ideally the entire organism, entire colony.
Roger that. You get
Well, he
enough,
comes with the territory. He can jump off if he wants.
Did you get enough beauty shots there before we?
Yeah.
Yeah,
took
that's
the
good.
sample?
If we could zoom on the squat lobster briefly, that is helpful sometimes.
Are we full zoom?
I don't know how much we'll be able to see.
Go ahead, push in.
Are we full zoom?
Let's find out. No, I am not. Now I am.
I'm going to slow your PRV down just a
Okay,
bit,
that's
Robert.
good.
Should help you.
That's good?
Thanks,
All right.
yep.
I'll get down to the base there again.
Mm-hmm. Great.
Yep.
You want to zoom out some or?
Oh, good. I
Good?
got two of other cameras on it there. I got one other camera. So I can get the whole rock probably if you really want to.
It's a big rock.
It's not that big.
No, that's a pretty big rock. Yeah.
2 fistfuls there.
We'll take the rock if you can get it.
You'll take the rock if I can get it? Did I hear yay?
That's a big
Yay.
rock.
It is a big rock, but we got a big bio box.
Underwater bonsai.
Was that an affirmative?
Sounds like yes from back row, yep.
Yeah, go wide and show me the whole rock, please.
Zoom out.
The rock and the animal.
Happy there?
Well, let me pick it up and see how it looks.
That's a big rock.
I like big rocks.
Maybe not the entire rock.
Well,
I think it's all or nothing, isn't
yeah,
it?
I think you're going to have a choice there.
let me pick it up and we'll see how it looks. You don't know until you get a hold of it. It's a big rock, all right. So the lasers.
Yeah, that's.
Oh, but we
that's
do have the squat lobster, which is
60
great.
centimeters across there.
You reckon?
Yeah. That's going to take every inch of a big box.
Yeah, I don't want to overload your sample box.
No, well, we
We'd have to put it on the side in the starboard box.
We typically, you know, once you get a sample in one of the boxes, unless you want to mix your samples, that's it. But
Lori, what would you like?
we can have a look and see if it'll fit. If it doesn't fit, we can.
It's going to be every bit of that box,
Okay.
I think.
Nobody needs the rock.
Roger. Okay, I'll put the rock back where I found it. Thanks for letting me have a go, though. I like to pick up rocks. Little kid in me is just screaming in joy right now.
Looking more like Chrysogorgia flavescens now.
You want to zoom in again?
That's fine there.
All right. That's good.
To try and keep the squatty, maybe it would be good to put it in the forward.
Sure.
If it doesn't run away.
If I'm way jet, we'll stay in there.
Usually they self-eject. Yeah. Oh, yeah, there's one at the bottom.
All right, can you zoom out?
When sampling squat lobsters, we have to be way quiet. They run away.
Wow, they are attached to their home. Beautiful.
Stand by one, Robert. And
Shoot. Sorry, I'm trying to figure out your starboard forward box open. Are you ready?
not quite.
You want to tilt down or
you'll have to rack back and show me the top of the box for opening there. I'm not sure where I am.
Track.
You can go wide, Jacob. Or Dave, whoever's over there.
And the box open?
Sure. I think we have one in the port side, so I'm going to have to go for starboard here. It should close a little bit, Robert, and see if I can.
Are you okay with that?
Wait, let me get the bushy bit in there first. Okay, close just a touch. And hopefully it will sink. Let go, craft. I'm going to stuff it in there a little bit.
bump it. You want to come off the box there?
Roger. Off. I'm out.
Lovely.
Nice.
That's the front oil box.
With 2 chirostylid question mark maybe spot lobsters.
Yeah, I like your box here, Dan. Good.
Yeah, you got control of all the cameras actually. You can
See.
zoom in and out on the upper camera as you wish. And I usually put it at mid-zoom. It gives you just the porch view there. That's where it's at now. I stuck it there on the...
All right, we're out of here. Moving on.
Yeah. I'll have to remind Lynette. I didn't hear you.
Bridge now.
There's a big sponge coming up in the upper camera.
30 meters, 085, please.
Can we zoom in again?
Thank you.
Going in.
Hey, Lynette, can you I just remind you and the new navigators, while we're sampling, we lose track of what's happening with Atalanta. Like we totally get focused on the sample and we forget Atalanta's like, slamming into the seabed or running over Hercules or whatever. So it helps us a lot if you keep an eye on that and what the delta is, and if you see that going out of the box, yell at us, because I forget about it all the time. I can't hear her at all.
You can't? She's being quiet.
Me?
Yeah.
That's because I'm muted.
No,
it's because I'm not talking to you.
the one next to you isn't.
Mind your own
because it's none of your business.
ROV, you want me to pull wide?
Roger.
Oh yeah, zoom out. Sorry. Unless we want to look at this, zoom in.
Yeah, let's have a look at that. Is that a grenadier? Yeah. Or McToten, or a
Sorry,
rat tail.
I gave you a tug there. I'll come back down.
Oh, wow. Beautiful.
Yeah, rat tail.
That's a, rat tail or grenadier.
Grenadier, yeah. A macrurid, is that what they are?
Yeah, macrurids, yeah. Lovely.
We're getting yanked a little bit. Oh, sorry.
Yeah, that
come down, Robert. Sorry, my bad.
It's probably running for the hills now.
Nice. Pointy.
Lovely. That's great footage.
All right, moving on.
Sounds good.
Yep, moving on.
Pull wide, ROV?
Go wide.
Yeah, go wide again. Is that another bamboo or is that a C-pen?
It looks
Zoom in?
branching, so probably
Zoom in?
bamboo-y. Yeah.
More of the same.
Yep. Bamboo.
Zoom out. little pink blob there. Can you zoom in again?
Pushing in.
What are you? Sea cucumber?
Yeah. Well, you might guess. All right, zoom out.
Come on out.
There's another one of them.
Another black coral on the side. Maybe a cup coral on the right there.
You want to zoom in?
Zoom in.
Sure. It's like a little sea anemone, doesn't it?
Yeah, I can't tell if it's the top of like a flabellid cup coral. It looks a little like a...
Yeah, it's got, looks like a septa there, the white.
Yeah.
Yeah, it is a coral. Scleractinian coral. I'd get a good view of that if we could.
Yep.
Lovely.
Got any more rope to give me?
Nice.
Oh, that's really good. That's beautiful. Nice picture.
Got it.
Wonderful.
All right.
You're getting run over.
There's that same coral again. We tested.
Yeah, hard. It's hard.
Yeah, we don't need to test another one. No.
You're right under me now.
All right, I'm going to zoom ahead a little bit. Can we zoom back out?
Yep.
Shrimps.
You're okay, just letting you know. Pretty benign out there. So.
Is that a crab with an anemone? Wait, can you zoom in?
Yeah.
Zoom in, video.
Zooming in.
No.
Yeah, it's a hermit crab.
Yeah, it is a crab with an anemone.
With something growing on him.
Yeah. All right, zoom out.
Okay, now you're behind the group.
Zooming ahead.
Yeah.
Can't zoom ahead, there's too much cool stuff to look at.
Yeah.
Big boulder.
It's a nice rock.
We're almost at the end of that
Goniasterid
move,
sea
so
star on
hold
there.
the boat for a while for you.
Are we still moving?
Oh, you got 5 meters left, you're good. We'll let it run out and let you get back out.
Yeah.
Do you want to look at that coral?
Interested in zooming on the coral?
Yes?
Yeah.
Okay. Can we zoom in?
Zoom in.
Paramaricid, maybe?
Sorry.
Sorry, I gotta come up. Give me a minute, let me come up a few meters.
Yeah.
Oh, you're landed. I'll come up slow down, slow. I was getting down to single digits there on my altitude because I'm closer to the hill than you are.
All right. Let me get on the other side of this rock instead of under it.
There you go. I can come back down a little bit more. I was getting close to single digits there.
Where'd it go? Where's it at? Up.
Upper cam is your friend.
Please zoom out again.
Zoom out.
I lost track of where I'm at.
Down and to the right.
Why is it not moving? There it goes.
I can speed it up a little.
Sorry, it's going.
Like some Chrysogorgids on the left, maybe?
Up at the top.
All right,
Top
can we
left.
zoom back in again?
Pushing in.
I had the down a little slow there.
Dan, I know you're otherwise occupied, but have you noticed, since you pay attention to the DSC cam sometimes, it go just extremely blurry all the time for no reason before?
The Thermocam?
Yeah.
If it's in autofocus, it will.
Oh yeah, it's in autofocus. The image, even the image on the screen all the time is blurry when I'm not taking a picture. Not sure.
Put it should be a manual focus.
Yeah, I tried that as well.
You got more zoom? What's this critter up in the corner there?
Got a Hermie.
Yeah, so it's moving in the current, so it's a soft coral. Yeah, nice hermit crab.
Okay,
there was
Robert,
a
you gotta get on with it. You're about to get
there was a stalk crinoid up. If you zoom out,
zoom out.
A little higher.
We gotta be kind of quick about it.
Yeah.
Where at?
Yeah, I don't know where.
All right, I gotta catch up here.
Lori.
Yeah, well, we could move the boat back if you want to look around here.
No, it's all right.
What's happening, Pat, is Atalanta's more to the east than Hercules, so Atalanta's single-digit altitude at the moment, which is
verboten.
officially breaking the rules there. And I'm gonna
oh yeah, it's just going out of frame at the bottom. That's
oh yeah. Backing up can get ugly too because you kick up the dirt.
Yeah.
Like where at?
For the pilots, how steep do you think that slope is?
hard to say unless we turn sideways to look at it.
Is this what we're looking at right dead ahead in the view?
Yeah, right in the middle of the view.
Zoom in again.
Pushing in.
You can, if you didn't fail math horribly like I did, you can look at this trend, this plot, depth plot trend here over time, and work out how fast we're coming up over
probably.
And
like
let's do it if you can.
What are we doing?
We'd like to collect that stalk.
Oh, we want to collect it. Okay.
Yeah, if we can.
Are we settled out?
okay, Lynette,
Yeah,
20 west,
settled.
please. 20 west.
Oh.
Again, it looks like it's growing on a rock. Maybe if we could, grab the rock.
Can we
there, Robert?
zoom back out? Yeah, I